We all know what a pill looks like, right? Small, kind of
Yeah.
unassuming. But I bet most people don't think twice about what it actually takes to make that medicine, to manufacture it on a large enough scale so it can actually help people.
Yeah.
It's pretty amazing when you think about it, you know?
Yeah.
There's so much science and engineering involved. It's a crazy puzzle to solve.
It is.
And, we love to dig into these things and find those aha moments. So today we are going deep on a real-world example straight from the world of pharmaceuticals. We're going to see how scientists and engineers took on this really intricate challenge of producing a complex molecule, but in large quantities.
Exactly. We're basically getting a behind-the-scenes look at how pharmaceutical process research and development works.
OPR&D.
OPR&D, yeah. I think of it like this. You have this promising molecule, right? It's discovered in the lab, and then it has to make this huge leap. It starts as this tiny little sample, and somehow it's got to become something that can be produced in kilograms. Kilogram quantities. That's what you need for clinical trials, and eventually, if everything goes well, for patients to actually use it.
Okay, so that's the journey we're looking at.
Yeah, the journey. And it's not always a smooth one, but it's full of clever solutions, and that's what we're going to unpack today.
So OPR&D, that's it's the brains and the actual work, I guess you could say. Figuring out the absolute best way to make a drug substance, the active ingredient, right? The part that actually does what it's supposed to do.
Absolutely. And it's not just about discovering it, it's about how you actually make it. Like, how do you design a manufacturing process that's reliable, that you can scale up?
Right. Yeah.
And that's where the real challenges come in, What works perfectly fine in a little flask in a research lab, it might completely fall apart when you try to scale it up to these giant industrial reactors.
I see.
So you got to problem solve. And that leads to some really innovative chemistry and engineering.
Yeah.
We're actually looking at a specific case study from the literature where we can see all of this unfold, the challenges, the troubleshooting, all the clever tweaks they had to make to the process. It's all documented.
Okay, so let's get into this case then.
All right.
So the goal at the beginning, it was pretty straightforward, at least on the surface.
Mm-hmm.
They wanted to produce kilograms of this pretty complex molecule.
Yeah.
And obviously, they weren't doing this just for kicks, right? This was a crucial step to get enough of this material to see if it could actually work as a medicine in clinical trials. So the stakes are pretty high.
Absolutely, yeah.
So they had to start somewhere. And they started with an initial synthetic route, basically a step-by-step recipe for building this molecule, chemically speaking.
Right.
And in this case, the original recipe was pretty involved.
It was quite lengthy.
Lots of individual steps,
Yeah.
and each one required specific conditions, specific temperatures, maybe a catalyst.
Yeah.
And a lot of times they'd end up with these intermediate compounds that they had to isolate and purify before moving on to the next step.
Yeah, it's like a really complicated Lego set, Like,
Yep.
you have all these separate stages and you got to do them in the right order.
Right.
And if you mess up one piece, well, the whole thing might fall apart.
So there's this one early hurdle they came across, and it really shows just how much detail goes into problem solving in this kind of work. They needed to do this very specific chemical transformation called the reduction of an aryl ketone to a methylene compound.
Yeah.
Now, I know that sounds like a whole lot of jargon, but imagine trying to remove a very specific type of connector from a complex structure. You have all these different tools you could use, and some are going to be way more effective than others.
Yeah.
And some might even damage the parts around the one you're trying to remove.
Right, exactly. And in chemistry, there are actually a bunch of different tools or methods you can use for this kind of reduction.
Okay.
You've got things like the Clemmensen reduction, the Wolff-Kishner reduction.
Right.
You could even use hydrogen gas with a catalyst or try a combination of other chemicals. And they all have their pros and cons depending on what specific molecule you're working with. So finding the right method or optimizing the one you've already got, that's a big part of the challenge.
So in this case, their initial approach for that reduction step, it involved using two really strong acids together, trifluoroacetic acid and sulfuric acid.
Mm-hmm.
Now, on paper, this might have seemed like a pretty straightforward way to do it, maybe even a shortcut, but when they actually tried it in the lab, different story.
Yeah, it turned out to be much more difficult than they thought.
This acid mixture ended up being incredibly corrosive, like way more than they expected.
It was a real problem.
It's like using a tool that's way too harsh for the job.
Yeah, it can damage the parts you want to keep.
And on top of that, They were getting a really low yield of the molecule they wanted,
Right.
which is a big problem when you're trying to manufacture something,
And
right?
it is. Low yield basically means you're not getting enough of your product at the end of the reaction.
Okay, so it's inefficient.
Very inefficient. It's like baking a batch of cookies and only half of them come out edible.
Right.
You've wasted a bunch of ingredients and a lot of time, and you don't have enough of what you actually wanted.
Makes sense. So that low yield with the corrosive acids, that was a big red flag.
It was a clear sign that they needed to either find a way to make that specific step way more efficient or come up with a whole new approach for that part of the molecule.
So they first tried to optimize the route they already had,
Right.
figure out if they could tweak the conditions,
Yeah.
maybe change the temperature, the amount of acid they were using, the timing of the reaction, stuff like that, just to see if they could improve that problematic reduction step and get a better overall result.
Exactly.
But they also had this other strategy they were using to boost efficiency
Oh, yeah.
called telescoping.
Mm-hmm. Telescoping steps.
Now, and I got to clarify, we're not talking about astronomy here.
No, not looking at stars.
So in chemistry, telescoping is kind of like streamlining a production line,
Right.
like in a factory. It means taking multiple steps in the chemical synthesis and smushing them together, basically.
Yeah, combining them. So you end up with this single continuous process, and you don't have to isolate and purify the intermediate products between each step.
Okay.
So it's like fewer stops, fewer transfers, saves a ton of time and resources.
It's kind of like instead of building a car by first assembling the engine, then moving it to another station to put on the wheels and so on,
Yeah.
you just try to do as much of the assembly as you can all at once.
Yeah, that's a great analogy.
So there are a few good examples of this in this case study. Like they realized they didn't actually need to isolate this one intermediate molecule in an end-tridyl derivative.
Right.
And just by changing the solvent they were using for that reaction, switching from dimethylformamide to dichloromethane, they could go straight to the next step without any purification in between.
Yeah, just like that. Just a solvent switch.
Yeah.
And it made a big difference. Got rid of a whole unit operation, made the process simpler, and they had to handle less material overall.
Makes sense.
Another cool example was with this reaction called Mitsunobu reaction.
Okay.
It's a pretty common reaction, but usually you'd have to purify the product afterwards.
Right.
But in this case, they did something clever. They followed the Mitsunobu reaction directly with a biphasic hydrolysis. It basically means they use two liquid layers that don't mix.
Okay.
And after they neutralized the whole mixture, they could just pull the amino acid they wanted straight out of the water layer. They didn't need any chromatographic purification at all.
And avoiding chromatography, that's a pretty big deal, right?
It is.
I always picture chromatography as these huge, complicated machines.
Yeah, they can be.
It seems like it would really slow things down if you're trying to produce things on a large scale.
Oh, absolutely. While chromatography is great for purification, it can be super time-consuming and expensive.
Yeah.
And it doesn't always scale up well to industrial levels. So if you can avoid it, especially when you're manufacturing large quantities, that's a huge win. It saves time and money.
Yeah, I can see that.
Makes the whole process much more efficient.
So it seems like they were making good progress with these telescoping steps,
They were, yeah.
making the whole thing more streamlined.
Mm-hmm.
But if I remember correctly, they were still stuck on that aryl ketone reduction.
Oh, yeah. That one was a real headache.
It was like this one stubborn problem they just couldn't shake.
Even with all the other improvements, that step with the strong acid mixture was still giving them trouble. Low yields, corrosion, the whole 9 yards.
It's funny, isn't it? Like you think you've got this well-defined process,
Right.
but then one tiny step can just throw everything off when you try to scale things up.
It happens all the time. And that's where the real detective work of OPR&D comes in. You got to identify the problem areas and then figure out how to fix them. Sometimes it's a small tweak. Sometimes you got to completely rethink your approach.
And that's why this whole process development thing is so important in the pharmaceutical industry.
It's crucial.
It's not enough to just know how to make a molecule in a tiny little lab vial.
Nope. You need a process that's robust, reliable, and scalable. Something that can churn out high-quality material in the quantities you need for clinical trials, and then eventually for the patients who need the medicine.
You're basically translating a scientific discovery into something real, something that can actually help people.
Exactly.
So what we've seen here is a real-life example of just how complicated it can be to manufacture a complex molecule, especially when you're trying to go from milligrams to kilograms.
It's a huge jump.
Yeah, and we saw how they used telescoping to combine and simplify steps, which made the whole thing more efficient. And they even managed to avoid some of those tricky purification steps, like chromatography.
Right. But then you have those stubborn challenges like the aryl ketone reduction, where the original method just didn't work at scale.
Yeah, and that's a good reminder that process development is an ongoing thing.
Oh, absolutely. It never really stops.
It's a cycle of finding problems, investigating them, and then tweaking and improving the process based on what you learn.
It's all about learning and adapting.
And it really makes you appreciate all the work that goes into making a single pill.
It does. It's easy to forget that behind that tiny tablet, there's a whole team of people who've poured their expertise into making it a reality.
Chemists, engineers, analytical scientists, they're all working together to figure out how to manufacture these medicines. It's a huge collaborative effort.
And it's just as important as the initial discovery of the drug itself.
Absolutely.
So, something to think about. Next time you see a pill, think about all the hidden obstacles and clever solutions that went into making it.
Yeah, all those unseen challenges.
And if you're interested in learning more, definitely check out the field of pharmaceutical process chemistry and OPR&D.
Yeah, it's a fascinating area where science and engineering meet to solve real-world problems, and it leads to medicines that can actually make people's lives better.
Couldn't have said it better
Yeah,
myself.
it's amazing stuff.